welcome to the Bluegrass, Kentucky. My name is Yang Jiang, a co-organizer for this workshop. I'm head of a aging brain and cognition laboratory in behavioral science department, University of Kentucky College of Medicine. It is my great honor and pleasure to introduce today's first speaker, Dr. Aina Puce. Dr. Puce is an Eleanor Cox Riggs Professor in Psychological and Brain Sciences at Indiana University. She has been a highly cited social neuroscientist who studies brain bases of nonverbal communications, such as perception of faces and biological motion. And very importantly, she made a great contribution to our field by leading the charge of champion the best practice for EEG and MEG studies. So I wanted to let you know that fresh off the press in Nature Neuroscience, so she has just published a guideline and she organized leading experts from all over the world for organization of human brain mapping, and they came up with this guideline, and they worked for a couple of years. And so it is, we're very fortunate to hear directly from Dr. Puce in her charming Australian accent on the topic of reusing EEG data and maximizing its value. Professor Puce.
Thank you, Dr. Jiang, and thank you for that lovely introduction. And let me just say two things related to that. That committee for Organization for Human Brain Mapping that I chaired with Cyril Pernay, and Cyril is the first author of that Nature Neuroscience perspective that we wrote. And so do check it out if you are interested in best practices in MEG and EEG. The second thing I wanted to say is, yes, I study the brain basis of nonverbal communication, and there's a delicious irony here, because our lab studies gaze detection and changes in gaze detection. And I realize now, as I present this talk to you, I can't look at any of you in the eye, because if I look at the images of you, you're not seeing me looking at you. If I look at the camera and you think I'm looking at you, I'm actually not. So there's something weird about this whole interaction with Zoom, and I'm painfully aware of that. So I'm going to try and make the best of it today. And I'm going to tell you about two studies that I did with colleagues in Paris, in France, and where we were reusing data and trying to make the most out of it, squeeze the most that we possibly could out of it. This is how I'm going to structure my talk today. I'm going to talk a little bit about the context for these studies and why we did what we did, describe the two studies, and then, of course, do a summary at the end. So I just want to put out a couple of thoughts that I've had going around in my head for probably the last 4 years or so. And first of all, I think is a need to focus on solid experimental design and think about data quality and adequate signal to noise and data code and sharing. And, best practices rock, okay? And I don't need to say any more about that because we've already talked about that. I think we need to integrate in a better way brain and behavior data from multiple methods. And this is value added. The sum of all of that greatly outweighs the parts. It's much more informative. It provides different data sets, provide context for another data set. And then finally, I make a plea. Let's please put the brain back into the body. And in the 20th century and at the end of the 20th century, we were all busy tearing the brain apart and looking at all the different areas and what their functions were. And we forgot about what it is that we do as human primates. So let's put the brain back into the body. And let's also get continuous measures of behavior. We have rich data sets, spatially and temporally rich data sets in fMRI and MEG, EEG. Let's make our behavioral measurements richer as well. So that's kind of the preamble for these studies I'm going to tell you about. So the first study that I'm going to talk about is the ability to try and detect signals at the sensor level. And I don't think that people think about this enough. So this is a study that was led by Maximilien Chaumont and is done also with Nathalie Georges in Paris at the Institut du Cerveau et de la Moelle Épinière. I was very fortunate to spend 6 months there on a sabbatical a couple of years ago. And this is work that we've continued and it's taken on a life of its own in a way. And Max led the charge on this. And this is an investigation of statistical power and how it impacts planning MEG studies. And it involves data reuse. So what we did, and let me take you through the methods, and sorry about the busyness here, but we are doing a methods workshop. We took, or Max actually, he did all of the backbreaking work here. Max took the Human Connectome Project MEG resting state data set of those who had a full sample. There are 89 of those 96 people. Whoopsie. He pre-processed and used the pre-processed data, that is, with the HCP pipeline. And of course, this was associated also with those subjects' structural MRIs, high-res structural MRIs, and their 3D sensor positions and their fiducial maps, of course. And then he created a single shell conduction model using field trip, using also another set of data from the HCP pipeline, the free surfer analyzed segmentation of the cortical mantle for each subject. This resulted in a mesh topology of about 65,000 vertices for each subject's brain. And then with using just a forward modeling type situation, created a dipole source of that strength that you see there systematically in each of the respective vertices in each of these meshes in all the subjects. And we looked at two different source models. One where the source dipoles themselves, the single dipoles in a vertex, were either constrained to be orthogonal to the individual's cortical mantle, or they were free from anatomical constraints. And what I'm going to do is, I don't have time to talk about all of the simulations. I'm just going to talk about the anatomically constrained ones today, and with the hope of showing you how complicated this story can be. Then what we did with that was Monte Carlo simulations, and we did different numbers of trials and subjects. So sampling without replacement, and then a trial was 25 milliseconds long. Remember, this is a simulation of time segments from the continuous resting state data, at least 2 seconds apart from one another. And then we split the trials into two sets. One set had this added signal, the dipole. The other set did not. We averaged the data across time points and trials separately for each set. And we did paired t-tests between the two sets across subjects at each sensor and noting their significance. And then did a cluster-based comparison, cluster-based correction, a method for multiple comparisons, well-known method. And then made sure that the peak.
First of all, we celebrated, right? So the temporal resolution is.
Okay. And then we did a comparison to make sure that the peak sensor, the sensor that was had the absolute value of the projection of the source signal, was included in that cluster. And we repeated that procedure 500 times for each trial by subject number pair that we had, and there were many of those. And then we noted the number of times the comparison was significant in 500 simulations. And then we examined the data across 2 dimensions: variability in position of the dipole and variability in orientation of the dipole. And as you'll see, there are some interesting ramifications of that. But to start us off, let me give you an example. 50 trials, 25 subjects. Pretty typical kind of manipulation, experimental study and in MEG. And this here shows what happens with these calculations, with a determination of statistical power, as I described, across all areas of the brain here. And you can see how it is not even. And you wouldn't expect it to be even, because we're talking about MEG, and in particular here, look at the mesial wall, very well favoured, even though the detectors are far away from the tissue. Think about it, because the neurons are, the dipoles actually in this case are facing in either this way or this way. The field is going around in this way. You've got a nice signal that's being able to be detected outside the head. Not so much here on these cortical surfaces, even though they're close to the detectors. Now you've got the neurons, or the dipoles, I keep saying neurons. The dipoles are oriented this way, perpendicular to the cortical mantle, and the field is going around this way. So you're not going to detect the signal as well. And so, this gives us pause for thought. Think about network analyses and different nodes activating, that we're not going to get equal signal to noise out of those necessarily. So let's put that in our heads, and I'll come back to that later. So let's have a look at some of these simulations and how they play out. We have let's choose one region, the fusiform gyrus, a highly studied area in face processing, one of my favorite subjects. And we chose this particular area because it has fairly low cross-subject variability in position. And so this is a lateral view of the brain showing the various dipoles in the 89 subjects and the net dipole that you get. This is magnified. This is the same view magnified here. This is showing it from a coronal view, and this is the axial view. I'm not going to dwell on the middle parts of this figure, but what I want to draw your attention to are these bottom the bottom row here. This is how this activity plays out across the sensor array on the head. Okay, left fusiform gyrus source, and you have a left-sided topography here. Now, look at simulations. Number of trials here as a function of the number of subjects. And this is the ability to detect the signal. And look at what happens. And as you'd expect, more subjects, better ability to detect signal, more trials, better ability to detect signal. And our example that I just started off with, 25 subjects and 50 trials, is about here. And we're getting a pretty good detection of that, greater than 80% from these interpolated lines here, contour lines. So that's great. Here we have a source that's far away from the detectors. Things are looking pretty good. But we've got low cross-subject variability there. Let's have a look at something where things aren't so good on the variability aspect. This is a source on the superior occipital gyrus, quite close to the detectors relative to the last source that I showed you. Here is the magnified view of that. And you can see the variability in the detectors. You can see the net vector not as strong this time. And why should that be surprising, given this kind of schmozzle that we see here. So here is the how the activity plays out across the sensor level, at the sensor level. And here is the power plot. Not so good, is it? So this can be, these kinds of simulations can be somewhat surprising and also illustrative and make us think about what we're trying to measure. So that was on the position variability side. What about choosing two sources that are a little bit more homogeneous in orientation? And to do that, we chose one source here in the insula. And the insula, of course, is an area that there's a lot of interest now. This is the magnified view of that. Here is the axial and here's the coronal view. And this is how the activity plays out in at the sensor level. And this plot here, it's quite possible to detect activity from this region. Not as well as from something like, say, the fusiform, but certainly possible if you're savvy about the number of trials and the number of subjects that you use. Certainly possible to do that. But now let's look at this one. And this is another area of brain that I'm very heavily invested in because of the work that I do with dynamic faces. And it's the superior temporal sulcus in its back end. Now, if I can find my mouse, here it is. I keep losing it. Here it is back here. This is the way the activity, these are the dipoles in the single subjects. Not a very large net dipole here. A lot of individual position variability. Why? Because this area of brain is highly variable across individuals. And not surprisingly, the signal at the sensors isn't seen all that well relative to the other things that I showed you. And of course, this power plot doesn't look so good either. So these are 4 examples that I've shown you where there are quite large differences in the ability to detect the signal. And they come about, there's variability across the head, and this just summarizes the standard deviation of the source position across subjects. And you can see it's not uniform, and particularly for the back of the head, it's highly variable. And that's unfortunate for those of us who study vision and study face processing like I do. This is a plot demonstrating that that variability in source position. There's all the sources across all of the vertices and a number of sources with these position deviations. And you can see the fusiform that I just showed you on that very first simulation plot sits here. And then the superior occipital temporal gyrus, occipital gyrus, which wasn't detected so well, sits out here. Same thing for orientation. It's not uniform in the brain, and you wouldn't expect it to be. But this plot just shows that to you, and this is how this has been calculated. And this is where the 2 examples that I showed you sit on our overall variability plot. So the insula, we could detect activity well. The STS, it was very poorly detected indeed. So to sum up the first study, we need to think about optimizing signal-to-noise ratio in MEG studies. And unfortunately, the bad news is that there is no single optimal data acquisition protocol, i.e., we can't say, okay, if you do X numbers of trials and X and Y number of subjects, you'll be just fine. You're not, based on what hopefully I convinced you that that's not possible based on what I showed you. Now, does that mean that we have to throw up our arms and give up doing these studies? No. I think we have to consider the potential activated structures in the experiment, the things that we're likely to activate, and design experiments with adequate signal-to-noise ratios for the least detectable brain structure. Now, if we can do that, then I think our analyses of functional and effective connectivity, things like dynamic causal modeling or graph analyses, can actually be meaningful and really deliver meaningful results. And then, of course, there's the question, how does this apply to EEG studies? And how does this apply to combined studies of MEG and EEG? And we don't know the answer to that. And we're going to be certainly looking into this further, because three of us are planning to work together. And we've had a lot of discussions about these questions. Okay, so I'm going to switch gears now and turn towards, again, data reuse, but not simulation, and using data that exists to try and do new analyses and try and squeeze everything we can out of the data. And this concerns the ability to track how information flows in the face processing system. This is work again conducted with in Nathalie Georges' lab in Paris, with Mariana Baborebolo leading the charge, and she's now at University College London. And this is the preprint. And so what we've done here is we've taken an existing data set that Nathalie had, an intracranial data set that Mariana and I worked on. And then we worked with Franco Pastilli's laboratory and his graduate student Dan Bullock to implement, they implemented a white matter endpoint calculation strategy, and then we combined the intracranial data together with these the white matter endpoint calculations that Dan had generated in Franco's lab. And this took a lot of work and a lot of time. So let me tell you a little bit about method, and again, detailed method slides here, not so much on results. We had, as I already said, we took an existing intracranial EEG data set with a fairly complex face processing task, which I'll tell you about in a moment. This was originally designed in Nathalie's lab by Josephine Huygen. And she extracted from that intracranial data set amygdala activity, electrodes in the amygdala, and published this paper in Social, Cognitive and Affective Neuroscience. And she has successfully gone on, and after her PhD, she finished her PhD, and this was one of her PhD projects. So we had this existing data set that had not been analysed. Nathalie had this data set and didn't have the people power to take this on. And so I was, when I suggested maybe popping over to Paris for 6 months for a sabbatical, this is what landed in my lap. And I was very delighted, and delighted to work with someone like Mariana Baborebolo, who really knew her neurophysiological stuff as well. So we first of all localized all the intracranial electrodes. Of course, it was MRI-based localization, and we did it by visualizing where the electrodes were ourselves. Mariana and I spent a lot of time because there was a lot of variation in anatomy in the posterior brain, so we spent a lot of time trying to localize that activity. We also spent an inordinate amount of time cleaning the data because the task was complex. We had these long epochs of EEG data. We started off, we had 18 subjects in the study, but remember, these were epilepsy surgery patients, and they were on a protocol where their medication was being withdrawn and they were waiting for seizures. So they had a lot of epileptic spike activity that was going on in their recordings. So much so that we actually threw out 7 of those patients out of our data set. So we were only left with 11. Of those 11 that were left, we only looked at the results from the correct behavioural trials. So we discarded the incorrect ones. And then we averaged data by condition in each site, in each subject. And for the aspect of the task, which I'll tell you about in a moment, we expressed these the activity in a bipolar fashion, taking recordings, re-referencing relative to the neighbouring contact on the depth electrode. We rechecked the anatomy, and then we normalised the evoked activity in every single person. Why? Because there is a lot of variation in intracranial activity. It depends where the electrode is, and it has nothing to do necessarily with the subject's brain so much. It's just a placement of the electrodes. So we don't want one subject's data to dominate the data set if we're doing group measures. So if we normalize, then everyone's on an even playing field. We then tested statistically that normalized activity. First of all, we had two steps. First of all, let's identify all the bipolar sites with z-scores greater than 1. And then those electrode sites that we identified, we did a cluster-based permutation test against 0 across time in the epoch at each site. And of course, we allowed for multiple comparisons. And then we visualized the activity as a function of 4 different ROIs. And I'm going to tell you, explain a lot of this stuff a little bit further, but that's just the big picture. Okay, let me tell you about the task. And this was the task that Josephine had put together in Natalie's lab. Complex task. First of all, there's a fixation cross, and then there's an onset of the face. And this face is a neutral face. The eyes look at you, and there are multiple identities of faces. And we call this face 1, the stimulus. After a period of time, this stimulus is replaced. There's no offset, it's just replaced by a face that shows an emotion, and there may or may not be a gaze change. So the emotion can be either happy or fearful. The gaze can either avert, and it can go to the left, to the right, or it can remain and stay direct. Everything's counterbalanced, of course, because there are a lot of combinations here. Everything's counterbalanced. And then finally, after a period of time, a little checkerboard target comes on the screen. The location of this target can be either congruous with the gaze or incongruous with the gaze. It could appear here. Or remember, there's a condition of direct gaze as well. So that could also be another condition. What's the purpose of this condition? From our point of view, nothing much. We want the subjects to focus their attention on the trial, and they have to press a button when one of these comes up. And there are catch trials. On about 12% of the trials, there's no checkerboard. They have to withhold their button press. So this allows us to check whether they're maintaining attention, whether they're doing the task properly. Because we want these other manipulations to be incidental and implicit. So that's the task. This is the 11 patients that we included in the study, pretty well distributed across gender. They're all adult subjects, mainly righties, just a couple of lefties. Seizure onset varied across the lifespan here. They're on a cocktail of anticonvulsant medications. Here are their seizure foci. And they were all assessed with this state trait anxiety inventory score, and they were all except one, and were all within normal limits. So this is where the intracranial electrodes are in all of the subjects. So we've color-coded them here. Mariana color-coded them on this particular diagram here in the 3 orthogonal planes, so you can see where they lie. And then what we did was, based on our anatomy and our careful looking at the anatomy, we divided the data into 4 regions of interest. And these are the number of electrode contacts in each of those 4 regions of interest. We have the inferior occipital cortex, the fusiform cortex, and the inferior temporal cortex, and the superior temporal cortex. Okay, so in the superior temporal cortex, we're including structures such as the superior temporal sulcus, the middle temporal gyrus. In the inferior temporal cortex, the inferior temporal sulcus, inferior temporal gyrus, Fusiform cortex, that's collateral sulcus, fusiform gyrus, occipitotemporal sulcus, and then the IOC, inferior occipital gyrus, and inferior occipital sulcus. And as you can see, the IOC brings up the rear, that has the most posterior sites. Here's the superior temporal cortex. It's going to lie dorsal to the inferior ventral cortex, which is also going to lie dorsal to the fusiform cortex. So that's where the electrodes are. Now, okay, I feel everybody recoil in horror when they see this slide. I'm going to take you through it slowly. These were the 4 regions of interest that I just told you about. This is their Y coordinate in M&I space. Okay, so the inferior occipital cortex, that brings up the rear, the back of the head. These other structures are very long, gyri and sulci, and they span quite a large part of the Y space. What we've done here is we've displayed the data in terms of their reactivity to the stimuli. Phase 1 is the face onset, remember. Phase 2 is the face that changes its emotion and gaze. Then we've classified electrode sites that have significant responses to both types of faces. And then there's a unresponsive category. Okay, so there are going to be sites that don't respond to any of those stimulus manipulations. These are the electrode sites in each particular region of interest, and they're color-coded accordingly. And then this is where those sites occur relative to their Talarak M&I Y coordinate. Now, the thing that I want to highlight your attention on is that for different areas of regions of interest, the proportion of activity, significant activity to the particular stimulus type was different. To explain this, if we look at the responsive sites in, let's say, the superior temporal cortex, the largest number of sites really that respond to the second, they respond to the second phase, that face with the emotion and gaze change, not so much to the face onset. There are some sites that do respond to both, but it's really more about the second phase. Let's contrast that directly to the fusiform cortex. Okay, so we've got a lot more sites, of course, here than in. We've sampled more than in the other cortex. But look at the proportion again. Now, when you look at the relative proportion of sites, the response to phase 2 isn't so great. So the balance changes. So then the question is, how does the balance change across these regions of interest? How does the balance of activity change? And how does that reflect with their known function? So, and this is another complicated plot. There's no way of looking at these data in a simple manner. Let me just walk you through this. This is a schematic, the lateral view of the head. Here's our inferior occipital cortical region of interest, fusiform, inferior temporal cortex, superior temporal cortex regions of interest. And what we decided to do was we remember we've got normalized data, it's bipolar, and we've decided to take a slice block that we created in here in 10 millimeter segments of that M&I Y coordinate, and that's represented here. So there's 6 slices that we've represented these active electrode sites on, and then this presents the averages across those 6 slices. And then these grey shadings here reflect confidence intervals, and the 2 face types are color coded here. Now, not every of these 6 slices that we've got, not everybody's got activity each ROI had activity in all of these. So you can see that there's 5 here and 5 here. And then the inferior occipital cortex, of course, that's behind this, that sits there on its own. But you can see that there's quite a variation between in the responses between the two faces. Oh, and then this is the number of electrode contacts in each location. So what we also did was we rectified the responses and if we needed to, and made a global average to try and make sense of all of this, because it's very dense data. So this is the face onset data. The first thing that happens in the system, the inferior occipital cortex has the earliest response, followed by the fusiform cortex and the inferior temporal cortex, and the superior temporal cortex brings up the rear. Contrast that now to this second one, face on the emotion and gaze face. Again, the IOC leads the charge, and now the other 3 areas follow it, but around about the same time. If we look at how these patterns of activity play out here, this is plotting the effect sizes across the 2 faces, and there will be larger effect sizes for the motion and gaze change in the IOC and in the ITC. But the point that I want to really bring your attention to here is look at the latency data. Here are the latency data. There are significant differences in these first 3 ROIs for favoring the onset of the face. That has the shortest latency. But now look at what happens in the superior temporal cortex. Now it's the depiction of the emotion and the gaze that actually drives the system much faster. So now what I want to do is unpack these differences in particular here, because remember, I haven't told you about emotion and gaze yet. I've just told you about the group of the response. So this plot shows the 4 regions of interest, color-coded electrode sites across all patients, with effect sizes for emotion and gaze in each ROI. And the one thing that comes out immediately is the gaze effect in the superior temporal cortex. Huge effect relative to everything else. And you can see the individual patients here and their effect sizes in these colored circles, and then these open circles are the group means. So this really stands out relative to everything else. So let's have a closer look at the waveforms here. These are all of the sites that had significant responses between the 2 types of gaze. Direct gaze is plotted here in red, averted gaze is plotted here in orange. And there might be some variability in the morphology of the responses, but the bottom line is that the averted gaze is always larger than the direct gaze. Where does this activity lie? These are the Y coordinates here, the M and I coordinates here, but look at where the activity is. It's always in the superior temporal sulcus. There's one in the medial temporal gyrus, and then it's all in the superior temporal sulcus. And these are all the electrode sites. This is all the data that I'm showing you. So the superior temporal cortex, we think, prefers these gaze aversions. How does this compare with the other stimulus dimension, the emotion, and the other ROIs? Well, this is what we call poster child, patient 17. We call him the poster child because he had nice sampling of electrodes in all 4 regions of interest. Not everybody did, but he's a good example for that reason. This is the data, some of the data I already showed you on the previous slide with direct and averted gaze. Now, look at the difference between the happy and fearful emotion. Not so much of a difference, is there? So it really is that gaze change that's driving activity in the superior temporal cortex. But now let's contrast the fusiform cortex to what we just discussed in STC. Not so much of an effect to eyes here, but in these statistical analyses, differences in the emotions in this region of cortex. So a different response profile. And the IOC responds to both. And the ITC, it's not that impressed by either of those. But there are very different response profiles across these 4 ROIs. So for those of you who do fMRI, think about what that might mean. So now we have our active electrode sites. How might the information be traveling in the brain? So to this, as I said, we turn to the expertise of Dan Bullock in Franco Pastilli's lab. And Dan had been working on a beautiful software routine to map out endpoint densities of fiber tracts. Here, what I'm showing you is Dan's plot of the results of taking 1,000 brains in the Human Connectome project and looking at tract endpoints of various known posterior brain white matter tracts and plotting one endpoint and then the other endpoint as a sort of like an overlay analysis. So this is the colors on the scale here reflects the number of subjects who actually were had a point endpoint for their tract in a particular location. So you can see here there's probably about 600 or 650 or so subjects who have that tract end there. So you can see the variability. And now you can compare where that is, where those tracts are relative to the active electrode sites. And to do that, we did an overlap analysis. So we've now plugged in our anatomical tracts. We've plugged in our 4 regions of interest with our face stimuli. We also added in the intraparietal sulcus. I didn't tell you about that earlier because we didn't have enough electrode sites to really do a statistical analysis, but we just put that in there as well, just to have a look in this analysis. So this is an exploratory analysis. The color coding here plots the proportion of intracranial electrode sites in a particular termination area for a tract. And these reflect various endpoints here. So the take-home message here is you don't so much look at these warm colors as much as you try and look at whether there are going to be connections between one end of the tract and the other. And that's the take-home message here, which doesn't come across very clearly here on the table, but let me try and unpack it for you. These are the tracts that we looked at for those of you who are white matter tract challenged, and I certainly am. And back here, it's a schmozzle of white matter tracts. From our data, we think that the inferior occipital cortex is actually sending the information to the inferior temporal cortex based on that latency analysis, but it happens via the inferior longitudinal fasciculus, and that the ITC then sends its information to the STC and the fusiform cortex via the posterior aspect of the arcuate fasciculus. We have these other dotted lines here that we think there might be some information traveling between, but we can't say definitively. So, okay, so what have we found relative to everybody else, and why is this important? This plot here is a plot I've stolen and modified from Cullinet Grill Spector's really lovely review in the annual reviews of neuroscience from a couple of years back about the known white matter tracts that link various structures in the visual cortex that are specific to face processing, such as these core areas in the face processing system, which I've drawn schematically for you here. And they're linked we know in fMRI studies in particular that there are other areas too, which are part of this extended system of face processing. You see them here, that also participate in these tasks. And the question is, how does this all work together? And so from the painstaking work of in Kalinek Grill-Spector's lab, and from work of many others, these white matter pathways are known. But what's not known is how these structures link to these other structures that we know are particularly important in face processing. And so let me give you a specific example. There is no known white matter pathway, direct white matter pathway between these two structures in the face processing network. People have looked at this and cannot find one. So back to our little modest little diagram, we think that the information going between the fusiform and the STS, that non-existent direct pathway, that information gets there through the inferior temporal cortex via this posterior arcuate fasciculus. So that's what's new in this study that we think we're adding. And it's up for grabs. This is a hypothesis, and hopefully that'll be interesting to people. People can test whether this in fact is true. So to sum up then the results of this particular study, the core and extended structures in face processing pathways were what we were trying to examine. We picked 4 regions of interest. We think that the IOC and that the inferior temporal cortex are important additions to the system that we really should be thinking about and focusing on. And the IOC, this idea is not new. Jim Haxby had proposed this originally. It's just nice to see it come to light here in this experiment so clearly with the latency effects that we see. The information appears to flow from the IOC and IOTC to other parts of the cortex based on these ERP peak latencies that we're talking about. But as I said already, we need to do some testing, and hopefully this will see some further studies by others to validate whether our claim is actually any even vaguely true. The other point that I want to make here is that latencies of evoked EEG activity, and particularly intracranial activity, I think have been underutilized for studying information transfer during, well, in this instance, face processing tasks, but that may be the case for other types of tasks as well. So, to sum up overall, where to from here? I think there's a clear need for further simulation studies of MEG and scalp EEG and the ability to detect sources at the sensor level. And I think we need to share simulation tools. And this was very apparent. I was at a meeting called Live MEEG this week, and so was actually Sylvain Bayet. We both spoke at that. And everyone is simulating wildly now. It's fantastic to see, and I hope people do more of it and really thinking about how to improve the results of their experiment. And people are sharing tools, and that's really good. And that's something that we're going to do too once we clean up our act as well, our code. And then this also brings up the question to improve other things. Let's improve our artifact detection and rejection methods. Maybe machine learning might be a way to try that. And these are exciting new avenues that we need to be thinking about. And then finally, we clearly need many more studies integrating information from different modalities. And I've given you an example here of hopefully how to integrate white matter pathway information with functional imaging data to map out information flow in brain networks. And this is ours is a pretty simple study. You could always be much more, you could make things much more complicated. This is an acknowledgement. This is an acknowledgement of financial sources. So Nathalie Georges, Max Chamon, and Mariana Baborebolo are all supported by a infrastructure funding from Investissement d'Avenir for their the CENIR platform, which is an MEG-EEG centre at the ICM in Paris. Vera Dinkelacke was a neurologist who helped us with identifying epileptiform abnormalities. She's funded by the John Boast Foundation in France. And I'm eternally grateful to my university who let me work on these projects overseas for 6 months with my colleagues in France. And I actually did more than just sit in cafes and drink coffee and go to museums, I think. And what's also good news out of this is we've kept working together, and it's now spawned a collaborative research grant in cognitive neuroscience, the NIH for us on this side of the Atlantic, and on the other side of Atlantic for Nathalie and Max. So with that, I thank you for your attention, and I'm happy to take questions.